Good morning, students. The topic we are going to see is plant tissue culture. The name of the lesson is plant tissue culture. See, in this lesson you are going to see how we are going to preserve the concepts, how we are going to perceive the concepts of the tissue culture, not preserve, it's perceive, and then the cognize the steps of the tissue. When you are going to list it out the tissue culture steps and the techniques, and understand the protoplasm culture, and then you are going to elicit secondary metabolites, and then learn about the plant regeneration pathway and the micropropagation, somatic hybridization, and lastly you are going to acquire the knowledge of patenting the biosafety and bioethics. Okay, so what is plant tissue culture? The definition for the plant tissue culture: the growing plant protoplast or organs away from the natural or normal environment. Normally how the plants are going in, not in that way, but it is done in the artificial condition, that is known as tissue culture. So growing the plant protoplast and tissues or the organs of the that are different from the natural or the normal environment, that is called as tissue culture. See, it is also known as, this tissue culture is also known as in vitro. In Latin, in vitro means growing inside the glass or in the test tubes. So in vitro meaning growing inside the glass or in the test tube. Now, before going into that, you should know about the meaning of the explant. What do you mean by explant? Explant is nothing but the transferring the living organism or transferring the living cells, a piece of the tissue which has been transferred from the animal or the plant to a culture medium or the nutrient medium, that is known as explant. So explant is that can be multiplied into several thousand plants in a short period of time and under the controlled conditions. So, okay, so what do you mean by explant? It's a piece of tissue that has been transferred from the animal or the plant cell into the medium, nutrient medium, where you are going to grow. That is known as the explant. This explant will multiply and it will grow into thousands of plants in a short period of time and the spaced under the controlled condition. Now, let's move to the tissue culture techniques. Now we will move to the tissue culture techniques. What do you mean by techniques? These are the techniques that you are going to follow for the culture. Okay. These techniques are useful. They are often used for the commercial production of the plants, for the commercial production and also for the plant research. The tissue culture techniques, why we are using tissue culture techniques is for the commercial production, for the plant research. Next, it's also an indispensable tool. Indispensable means important tool for the regeneration of the transgenic plants. What are transgenic plants? The plants that are grown by the genetically modified techniques. New plants, not by the natural method, it is by the artificial method. Inducing a gene into the plant and getting the variety that you want. That is known as transgenic plant. Next, due to that technique, what are the uses you are going to attain? Conserve the endangered species. What do you mean by endangered? Endangered means what? The species that are going to disappear from this world. Those type of species are conserved. Conserved means what? They are preserved. They are again grown. Next, the germplasm preservation. Next, the germplasm is preserved. Okay, and then the industrial production of secondary metabolites. So, okay, the plant tissue cultures conserve the endangered species, preserve the germplasm, and also for the industrial production of the secondary metabolites. Okay, now we are going to see who is the father who proposed this plant tissue culture. His name is Gottlieb Heberland. The name of the person who proposed this tissue culture is called as the name is Gottlieb Heberland. He was born, he introduced this type of totipotency or the tissue culture in 1902. He proposed that in 1902. By using the cells of Lamium purpureum. He is called as the father of tissue cultures. Gottlieb Heberland in 1902 proposed the totipotency he found using the cells of the Lamium purpureum. He is called as the father of the tissue culture. Okay, next we'll move to the basic steps of the tissue culture. How we are going to do the tissue culture? What are the things that is going to happen in the steps that are going to happen in the tissue culture? First one is the totipotency, next is differentiation, third is redifferentiation, and fourth is dedifferentiation. What do you mean by totipotency? Totipotency means from the small cells that is taken out, isolated, we are going to grow a new plant. From the cell, new big plant arises. That is known as totipotency, the potential. Okay, the property of the live plant that have the genetic potential when cultured in the nutrial medium to form a complete plant. A small cell has the potential to form a big plant, the new plant, that is known as totipotency. Differentiation means what? That is nothing but the biochemical and the structural changes by which the cells that form the function. Next is redifferentiation. Redifferentiation is the differentiation of already differentiated cell. One cell already differentiated, that is again, re means what? It is again, re means what? Again it is going to differentiate it to form the whole plant. That phenomena is known as redifferentiation. And dedifferentiation is reversion of the meristematic cells stage. Reversion of the cells, mature cells to meristematic cell stage, that is known as dedifferentiation. These are the phenomena you should learn, the basic techniques or the basic concepts in the tissue cultures. One is called as the totipotency, differentiation, redifferentiation, and dedifferentiation. I repeat one more time. Totipotency is the potential of the living cell to form a big complete new plant, that is totipotency. And differentiation is nothing but the process of biochemical and structural changes that forms in the function. Then redifferentiation means what? It is the already differentiated cell into another type of cell, that is known as redifferentiation. Re means what? Again it is going to differentiate it, that is called as redifferentiation. And redifferentiation is nothing but the reversion of the mature cell, reversion coming back to the meristematic states, that is leading to the formation of calyx, that is known as redifferentiation. Okay, now we will see, we are going for the plant tissue culture, that is given as PTC. I have used the short form as PTC. As a continuation of the last chapter, I am taking this lesson. There are fundamental principles at the back of the plant tissue culture. That is nothing but it is grown in the aseptic condition. Aseptic means without any contamination, in a pure form, in a clean environment. That is called as aseptic. The plant tissue culture is grown in the clean environment, that is called as aseptic. And in vitro means what? Inside the glass tubes or inside the test tubes. This plant tissue culture is grown inside the test tubes. And next, the fundamental principles are the part being removed, that is called as the explant. That explant is selected and isolated from the plant, and it is taken out for the first step. And next step, it is the conditions that are physically and chemically that are maintained for the growth of the explant. Physically and chemically the conditions are maintained and controlled. Physical is nothing but the environment. Chemical is nothing but chemical is the nutrient medium. Okay, the physical means the outside medium, chemical in which it is growing, that is known as the environment, that is known as the nutrient medium. This is the nutrient medium and this is the environment, the surrounding environment. Both the environment should be maintained, the conditions should be maintained, and it should not be both the conditions should be regularly maintained. The physical environment, the outside environment, and the nutrient medium should be maintained. Okay, that's all about the plant tissue culture. Now we will move to the laboratory facilities for the plant tissue culture. What are the laboratories? What should be there for the plant tissue culture? A laboratory should possess to do the plant tissue culture. First of all, you should have the glass vessels. Those glass vessels should be washed. You have the washing facilities. You should have the washing facilities of the glasswares and oven to dry the glasswares. You should have the washing facilities to wash the glasswares and the oven to dry the glasswares. And the medium, you should have medium preparation room with, you should have the room with for the preparation room with the autoclave. Autoclave is nothing but the machine that is used to clean the vessels through steam. Okay, and then electronic balance and the pH meter. You should have a preparation room with the medium is what? The culture medium in which you are growing to grow the plant. That medium preparation room you should have equipped with autoclave, electronic balance and pH meter. And then the transfer area should be sterile. Sterile means what? It should be clean and clear. A laminar air flow and the bench with a positive pressure. And you should have the ventilation unit and it should have the HEPA filter. HEPA filter, this is a high efficiency particulate air in which it will be free from the bacteria. The filter is free, it will make the room free from the bacteria. And now the explant that is removed from the plant, that is grown into the culture tube at the temperature of 22 to 28 degree, and the illumination that is provided is 2400 LUV, and the photo period is for the growth of the plant is 8 to 16 hours. Photo period, it should be shown to the photo means what? To the light for 8 to 16 hours, and the humidity for the growth of the plant is 60 degree. We should not have more humidity, but when the humidity is high, it makes a tendency for the bacteria and other microorganisms to grow. I repeat, what the laboratory facilities. First of all, the laboratory should have the facility to wash the glass, oven to dry the glasswares, and the room should be in the medium preparation room, and where the medium is prepared, it should have the autoclave, electronic balance and the pH meter. And the transfer area should be sterile room. It should have the laminar airflow, positive ventilation, and the ventilation you should fix HEPA filters, that is high efficient particulate air. It should not have any bacteria. It should be in the aseptic condition. I told you before itself, aseptic is nothing but the without any contamination. It should be free from the contamination. And then the explant that is inoculated into the culture, it should be grown in the test tubes with 22 to 28 degree. The illumination is provided is 2400, and the required photo period in which it is shown for the light, inside the light, it is 8 to 6 hours, 16 hours, and the humidity is 60%. All these conditions are required in the lab for the growth of the plant. That's all about the laboratory facilities. Thank you.